can start.
Okay, start.
Hello, everyone. I want to thank everyone here for attending this amazing event, and I'm very glad and honored to be here sharing with you my research topic. My name is Fang-Chih, and my advisor is Dr. Yanfu Guo. I'm from the National Taiwan University, and the topic I want to share with you today is quantifying virus resistance level using convolutional neural network. Okay, so plant diseases is a major issue in agriculture. According to the Food and Agriculture Organization of the United Nations, plant diseases cost the global economy around $220 billion per year. The figures here are some examples of how different virus can cause local lesions on leaves of many plants. And I'll show you later of how these local lesions or like lesion spots on the plants can be significant. Okay, so currently there's no direct treatment for plant viruses. However, it can be prevented. Viruses are mostly carried and spread by insects in the field. So the current method of plant virus prevention is by eliminating these virus-carrying insects in the field with pesticide. According to the Council of Agriculture in Taiwan, 9,848 tons of pesticide is used in single year 2020, of which 355 tons are highly toxic. So apparently, the long-term and inappropriate use of chemical pesticides can be harmful for both human and the environment. So the studies have been conducted and shown to present a more novel and natural approach to plant virus prevention, which is by inoculating plant growth promoting rhizobacteria, or the PGPR, into a plant, and it is shown to be able to boost the plant's immunity towards virus. And this is proven to be able to reduce loss in the field. Okay, so no matter what approach we use to try to prevent virus from infecting our crops, one very important thing we have to do is to evaluate the effectiveness of our approach. So in order to evaluate the effectiveness of different approaches for virus prevention, certain index should be calculated. The plant disease incidence is one of the most commonly used index, and it can be defined as the number of plant units that are visibly diseased relative to the total number assessed. Or in our case, in simple words, the ratio of lesion spots on the leaf to the area of the whole leaf. So the three leaves here are of different disease incidence, which is low, medium, and high from left to right respectively. The disease incidence calculating is currently involving manually counting with the naked eye, which are exactly the problems modern agriculture faces. Labor-intensive under a labor-shorting industry, and time-consuming in a society that demands efficiency. So my objective here is to develop a local lesion identifying system based on CNN and computer vision. With the system, we will be able to separate the lesion spots on the leaf from the healthy leaf parts. Then I will convert the results to quantifiable data, for example, the number or area of the lesion. So to do this, here we're facing a semantic segmentation task. Different from classification task with bounding boxes, a semantic segmentation task classifies every pixels in the input image. You can see the demonstration figure here, like the cars, the traffic sign, the pedestrians are all classified and given a certain color. In our case, here we want to develop a model that takes a leaf with local lesion image as inputs and output a mask that segments the local lesions. Okay, so here I want to talk about my data set, my material. 34 images of a single quinoa leaf were collected and labeled, which these leaves were infected by the Zucchini mosaic virus. And my data set are divided into a training set of 30 pictures and a validation set of 4. 2 images were taken per leaf, and each image is the original size of each image is 3,456 by 4,508 pixels, which were scaled by 0.5 before being input into the model. The images were annotated by hand using the Label Me annotation tool. And as you can see, each local lesion were bounded by a polygon. The deep learning model we choose to use in our study is the UNet model. UNet model is the deep learning architecture that is commonly used in image segmentation tasks. It was developed by Olaf Ronneberger et al. In 2015. The architecture consists of a contracting path, which is responsible for capturing the context of the input image, and a symmetric expanding path, which is responsible for generating a high resolution output segmentation mask. The contracting and expanding path were connected by skip connections, which allow the network to combine low-level and high-level features from the input image to generate a more accurate segmentation mask. UNet has been widely used in medical image segmentation and is known for its ability to accurately segment objects in images. And that's why we choose this model as our deep learning model. So here are some training details for this model that might be interesting. This model has been trained for 3 epochs, with the batch size of 1, scaling factor of 0.5, initial learning rate at 0.00001 with learning rate adjustment, and we used the dice coefficient to evaluate the model's performance. Okay, so for the training result, the UNet model we trained achieved a dice coefficient of 0.6015. So you can see on the left that on the left is the original input image of a single quinoa leaf infected by the Zucchini mosaic virus. In the middle is the ground truth labeling that was annotated manually. And on the right is the model's prediction. So if you look closely, you can see that the results or the model tends to predict clustered lesion spots as one big local lesion. And this is one problem we're facing. Another problem we're facing is that the model tends to predict some false negative cases. You can see that on here and here. There are small lesion spots that the model think it is a lesion spot, but we're not on the ground truth labeling. And next, I'm going to share with you why I think these error occurs. So the first discussion or challenges that I want to mention that might have affected the model's performance is that, as I mentioned, 2 images were taken on the same leaf, but under different environments. So they may have different exposures or lighting, contrast, brightness, etc. And this might cause the inconsistency in labeling. So you can see these 2 pictures are of the same leaf, but they're labeled slightly different. So it might be inconsistent and confusing to the model. The second challenges we're facing is that there are more than just local lesions on leaves. There might be wounds, there might be bug bites, but not local lesion spots and should not be considered as one. So they look very similar to local lesions, and we can tell them apart by human eye, but not so easily for the model. So the model tends to predict the wounds or the bites as a local lesion, and that can cause the false negative occurrence. So to conclude my research, I want to say that I proposed a UNet model that was trained to identify local lesions on the leaf. The model achieved a validation dice of 0.6 with extremely small data set. And some future works that I think should be done. More labeled data should be acquired, and the consistency and accuracy of the annotation should be improved. I want to express my thanks to partners in the Taiwan Agriculture Research Institute and the lab members for their assistance in my research. Thank you for listening.
Thank you for your presentation. So from the audience, so do you have any questions? Yep. Yes, please.
Thanks for the nice work, for a nice presentation of your work. You say you have 34 images?
Yes.
And that means, and two images per leaf. That means 15
Yes.
leaves.
Yes, approximately.
Okay, so
yes.
that's quite limited data set.
Yes.
So you are planning also to extend that?
We acquire our data set from our partners. We collaborate with them, and we're kind of limited by the amount of data set they provide us. So we're currently working on trying to get more data set from them, but this is what we're having now, and I'm trying to do my best to make the most out of these limited data set. Yes.
Yeah, that's also my question. What is the variation in your data? How is the environment where you took the images? Is it very much controlled? And how do you think if you take other images from other partners, then I expect there will be a lot of variation in the lighting and the exposure time and all these kind of things, which might have effect on your either the labeling or the inference of your network.
Yes. So I think if we ever acquire more data set, the annotation must be much more accurately, much more controlled in order to effectively eliminate the effect of environmental issues. Yes. Yeah. Did that answer your question?
Yeah.
Yeah. Thank you.
Yeah. Thank you.
So one more short question, just short time.
Hello, thank you for a nice presentation. I think that at the end, this is a problem of color segmentation, actually. So have you tried to compare your method with a classical algorithm for color segmentation? That's my first question.
Yes, thank you for your question. And this is actually a very important question, that if my research is making difference with traditional image processing methods, right? Mm-hmm. So I've tried with image processing methods, and the main concern here is that, like I said, there are many wounds and bug bites that look very similar to local lesions on leaves. And in my experience, I'm not able to singly use the image processing methods to tell them apart. But I am hoping that a more complex deep learning model can do that. Yeah, but of course, we need more data set and more accurately annotated images in order to achieve that. Okay. Yes.
Thank you. This is related with my second question because
yes. Sorry, time.
Okay, no problem. Thank you.
Sorry.
Thank you. We can talk later if you want. Yes, thank you.
You can discuss later. So thank you so much. So the third presenter, we have to go to the next presenter, Hiroki Ichihara.